The Harbin skull was reportedly discovered in 1933 during the construction of a bridge over the Songhua River in Harbin, the capital of Heilongjiang province in northeastern China. Amid the turmoil of the Japanese occupation, the worker who found it hid the fossil in a well to prevent its seizure, and it remained concealed for decades. Ironically, it was hiding this fossil in a cold, dark well that is likely the reason that DNA is still preserved. If scientists had been given the skull in 1933, it likely would have been preserved with chemicals that killed the remaining DNA. Or worse, the fossil may have been lost during World War II, as was the Peking Man fossils. Only in recent years did the skull resurface when the family of the original discoverer revealed it to Chinese scientists. The fossil was immediately recognized as extraordinary due to its size, completeness, and unusual blend of archaic and modern traits. Because it lacked precise stratigraphic context, the skull's age and lineage were initially difficult to determine. However, uranium series dating placed it at more than 146,000 years old, squarely within the Middle Pleistocene. Despite these challenges, the specimen became a focal point for debates over human evolution in Asia, and was tentatively assigned to a new species, Homo longi, pending further genetic and morphological analysis. It is only now, with the recovery of mitochondrial DNA, that its true place in our lineage, as a Denisovan, can be confidently declared. For over a decade, Denisovans have lingered at the edges of human evolutionary knowledge, known more by their genetic signatures than by their faces or bones. Discovered initially in the frozen altitudes of Siberia's Denisova cave, this enigmatic human lineage left behind mere fragments, a pinky bone, a molar, and tantalizing traces of DNA in modern humans from East Asia and Oceania. But in 2025, a scientific breakthrough fundamentally changed our understanding of the Denisovans through a skull discovered in Harbin, China, and crucially, its mitochondrial DNA. The Harbin fossil, known as Homo longi, has now been definitively linked to the Denisovan lineage through sophisticated paleoproteomics and mitochondrial genome sequencing. This long-awaited connection not only affirms the Denisovan identity of one of the most complete archaic skulls ever found, but it also illuminates the broader biological picture of the Denisovans, revealing their likely body size, their evolutionary relationship to Neanderthals, and how their anatomy corresponded with their vast ecological range from Siberia to Southeast Asia. The Harbin cranium, unearthed near the Songhua River in northeastern China, had long perplexed paleoanthropologists. It displays a remarkable combination of features. An enormous brain case over 1,500 cubic centimeters, similar to or larger than modern humans, a wide flat face, a thick brow ridge, and exceptionally large molars. These traits defy simple classification into known Homo lineages like Neanderthals or early Homo sapiens. Previously, Chinese scientists proposed that the skull represented a new species, Homo longi or dragon man, but its taxonomic position remained controversial. Now, thanks to newly published studies, this mystery has been resolved. Chinese researchers developed a new method of identifying hominin populations using ancient proteins and DNA extracted from dental calculus, essentially fossilized plaque on teeth. Their success in recovering ancient mitochondrial DNA from the Harbin individual's dental calculus is a landmark achievement. Despite the high degradation typical of ancient DNA, especially outside permafrost zones, the researchers constructed multiple DNA libraries and painstakingly reconstructed the mitogenome. The Harbin individual's mitochondrial DNA carries Denisovan-specific mutations, linking it directly to other known Denisovan individuals, particularly Denisova 3, the well-preserved finger bone from Siberia. This not only establishes Harbin as a Denisovan, but also places it in one of the earliest branches of the Denisovan mitochondrial lineage. Mitochondrial DNA is genetic material found in the mitochondria, the powerhouses of cells, passed down exclusively through the maternal line. Unlike nuclear DNA, which is inherited from both parents, mitochondrial DNA provides a more direct and unbroken maternal lineage. It evolves relatively quickly and is often more abundant and better preserved in ancient remains. In the case of the Harbin skull, mitochondrial DNA not only confirmed the individual's Denisovan identity, but also provided crucial insight into how Denisovans spread across Asia. The Harbin skull dates to at least 146,000 years ago, pushing Denisovan presence in Northeast Asia much further back than previously documented. This wide distribution, from Siberia's Altai Mountains to the plains of northern China and possibly even Southeast Asia, suggests that Denisovans were one of the most adaptable and successful archaic human populations. The recovery of the Harbin mitochondrial DNA allows us to examine more deeply the relationship between Denisovans and their better-known cousins, the Neanderthals. Genetically, Denisovans and Neanderthals are sister lineages, having diverged from each other roughly 400,000 to 500,000 years ago from a common ancestor that also gave rise to modern humans. Despite their shared ancestry, the two species evolved distinct morphologies and adapted to different environments. While Neanderthals are associated with Ice Age Europe and parts of Western Asia, Denisovans roamed farther east. This eastward expansion may have exposed them to diverse climates and altitudes, from frigid highlands in Tibet to subtropical jungles in Southeast Asia. The Harbin skull, with its robust features and enormous size, supports the view that Denisovans had unique physical adaptations, possibly for enduring cold climates or consuming tough, abrasive foods, as implied by their massive teeth. Interestingly, the dental and cranial morphology of the Harbin skull aligns closely with other fossils from East Asia, such as those from Jinyushan and Dali. Some researchers now believe that these fossils may also be Denisovan or Denisovan-admixed individuals. The broad range and anatomical variability of Denisovan-like traits suggest a complex evolutionary scenario involving repeated migrations, interbreeding, and localized adaptation. One of the most striking inferences from the Harbin skull is its implication for Denisovan body size. The term body mass refers to the total weight of an organism, which in humans correlates with overall energy requirements, metabolism, heat retention, and biomechanics. In paleoanthropology, body mass is often estimated using cranial or postcranial metrics, such as femur length, pelvic breadth, or, in cases like Harbin, cranial volume and facial robusticity. The Harbin skull's cranial capacity of over 1,500 cubic centimeters places it in the range of modern Homo sapiens and even exceeds most Neanderthals. Its exceptionally thick bone walls, wide zygomatic arches, and enormous molars suggest a physically powerful individual, likely weighing around 100 kilograms, 220 pounds, and standing around 1.8 meters or nearly 6 feet tall, though this is speculative in the absence of postcranial remains. Such a body mass would align with the hypothesis that Denisovans, like Neanderthals, were cold-adapted hominins. Larger body mass in hominins is typically associated with climates where retaining heat is essential. In colder regions, a lower surface area to volume ratio helps conserve body heat, known as Bergmann's rule. This is evident in the Neanderthals' stocky bodies, and may also be true of Denisovans, especially if they inhabited Ice Age landscapes in Siberia and northern China. Additionally, larger body mass often correlates with greater muscular strength and physical robustness, traits likely necessary for survival in the demanding environments Denisovans inhabited. Hunting megafauna, building shelters, and enduring harsh winters would all benefit from greater size and strength. The Harbin cranium's enormous chewing muscles and broad face also suggest a diet requiring heavy masticatory force, possibly including raw tubers, fibrous plants, and large animal bones. Prior to this discovery, Denisovans were practically faceless. Reconstructions relied largely on DNA studies and comparisons with modern populations. But the Harbin fossil finally gives Denisovans a face, broad, flat and massive, and a genetic identity that links it to both East Asian fossils and modern populations with Denisovan ancestry. Researchers identified 122 single amino acid polymorphisms unique to hominidae species and 95 proteins from the Harbin cranium. 3 of these amino acid changes are exclusive to Denisovans and provide a direct link to the previously sequenced Denisova 3 genome. This molecular connection allows researchers to place Harbin within the Denisovan family tree, refining our understanding of their phylogeny and regional variation. The confirmation of Harbin as a Denisovan has sweeping implications for the study of human evolution in Asia. For decades, paleoanthropology in East Asia operated under the assumption of a linear progression from Homo erectus to modern humans. However, the Denisovan identity of Harbin challenges this model and suggests a far more intricate picture. Multiple overlapping lineages, regional variations, and extensive interbreeding. The Harbin mitochondrial DNA discovery also forces a re-evaluation of other fossils in China, especially those with ambiguous features that don't neatly fit into known categories. Fossils from Dali, Jinyushan, and even Hexian may now be revisited with an eye toward Denisovan traits. It's plausible that Denisovans were not a minor or isolated lineage, but rather a dominant hominin population in Pleistocene East Asia. Moreover, this research establishes a new method for extracting ancient human DNA from dental calculus, opening the door to studying many more fossils that lack well-preserved bones for DNA extraction. This is particularly critical for warmer climates where traditional DNA retrieval methods often fail. The Harbin skull has not just added a face to the Denisovans, it has transformed them from a genetic whisper into a living, breathing population with a geographic footprint, anatomical identity, and a tangible place in the human story. Its mitochondrial DNA connects it directly to Siberian Denisovans, while its sheer size and morphology hint at a powerful body adapted to harsh environments. More than just a fossil, Harbin is a window into a forgotten population, one that helped shape the genetics of millions of people today. The discovery reveals that the Denisovan story is not confined to a cold cave in Siberia, but sprawls across the vastness of Asia, waiting to be rediscovered in bones, teeth, and the hidden code of mitochondrial DNA. As paleogenomics continues to evolve, the Harbin cranium may be just the first of many ancient ghosts to rise from the depths of the Pleistocene and reclaim their place in our shared ancestry. The question remains, what happened to the Denisovans of China? Why do East Asians have so little Denisovan DNA compared to Neanderthal DNA? One emerging hypothesis is that Neanderthals didn't stop migrating at Denisova Cave and migrated into China after 100,000 years ago, absorbing and replacing the Denisovans there. Thanks for watching. Please subscribe.